Hello, dear students. Welcome to the e-learning platform of Sanjeevani Mahavidyalaya Sapoli. Here is Department of Botany, semester 4th and semester 6th, paper 9th and paper 14, plant metabolism, biochemistry and biotechnology for B.Sc second year and third year. In previous video lecture, we discussed on cyclic photophosphorylation, and now in this lecture we are going to discuss non-cyclic photophosphorylation or Z scheme. The non-cyclic photophosphorylation, it is a normal process of photophosphorylation. In this, the expelled electron does not come to the reaction center. That's why it is known as non-cyclic photophosphorylation. And that non-cyclic photophosphorylation is carried out with collaboration of photosystem 1 and photosystem 2, or reaction center 1 and reaction center 2. The electron released during photolysis of water is picked up by reaction center 2, that is P680. The same expelled electron absorbs a photon of light. The expelled electron has an energy is equivalent to 23 kilocalorie per mole. The expelled electron, when absorb photon energy, it is in excited state, and it is in the excited state it passes through a series of electron carriers or acceptors like pheophytin, plastoquinone, cytochrome B.F complex and plastocyanin. While passing over to cytochrome complex, the electron loses a sufficient amount of energy. When the electron expelled from the reaction center by absorbing photon energy, that expelled electron or ejected electron is accepted by number of electron acceptors, and each electron acceptor, the electron loses some amount of energy. And when the electrons comes to the cytochrome complex, sufficient amount of energy is loses or lost by the electron. Why the energy is lost, or what is the use of that loosed energy in the electron acceptors? That loose energy is used to prepare ATP. The electron handed over to the reaction center 1, that is P700, by plastocyanin. The reaction center 1 expel the electron After absorbing the light energy, the expelled electron passes through ferredoxin and NADP reductase. The expelled electron pass through the ferredoxin and NADP reductase, which combines with NADP and to form NADPH, or NADP is reduced to NADPH. NADP is reduced to NADPH2. ATP is not formed here, or ATP is not synthesized. Instead of ATP, here NADPH2 is formed. The energy released by the electron is actually used for pumping the hydrogen plus ions across the thylakoid membrane. The energy released by the electron actually used to H plus pumping across the thylakoid membrane, and it creates the electron or proton gradient. This gradient, proton gradient, triggers the coupling factor to synthesize ATP and ADP. That proton gradients triggers the synthesis of ATP from ADP and inorganic phosphate. That is the non-cyclic photophosphorylation. In the non-cyclic photophosphorylation, the expelled electron does not come to its original place. That's why it is known as non-cyclic photophosphorylation. Chemiosmotic hypothesis for this non-cyclic photophosphorylation is also suggested. How actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forwarded by Peter Michel in 1961 to explain the mechanism of ATP formation. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of proton gradient across the membrane. This time these membranes of the thylakoid. Here in the non-cyclic photophosphorylation, that membrane is thylakoid membrane. There is one difference through here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, the proton accumulates in the intermembrane space of the mitochondria when the electrons move through the electron transport system. But here in the thylakoid membrane, the electrons or the proton gradient created inside the thylakoid membrane, that is in the lumen. Let's see the causes of proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons and then transport to determine the steps that cause a proton gradient to develop. Since splitting of water takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water molecules, which accumulates in the lumens of the thylakoid. As the electrons moves through the photosystem, the protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electrons not an electron carrier but to an hydrogen carrier. Hence, this molecule removes proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into inner side of the lumen or lumen side of the membrane. How the proton gradient is produced. The electrons released that are found on the outer side of the membrane and the splitting of water molecules in inner side of the lumen or membrane. That electrons moves towards the protons with hydrogen ions, and because of that the proton gradient is produced in the lumen or inner side of the membrane. The NADP reductase enzyme is located on the stroma side of the membrane along with electrons that comes from the acceptor of electrons of photosystem 1. Protons are necessary for the reduction of NADP to NADPH2. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decreases in the number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads the release of energy. The gradient is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channel of F0 of the ATPase. The ATPase enzyme consists two parts. One called the F0 is embedded in the membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called F1 and produces on the outer surface of the thylakoid membrane on the side of that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the F1 particle of the ATPase, which makes the enzyme synthesis several molecules of energy packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATPase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATPase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATPase enzyme that catalyze the formation of ATP. Along with NADP is produced by the movement of electrons. The ATP will be used immediately in the biosynthetic reaction takes place in the stroma, responsible for fixing carbon dioxide and synthesis of sugar. Where are the ATP and NADP used? We have seen that The products of light reactions are ATP, NADPH and oxygen. Of these, oxygen diffuse out of the chloroplast while ATP and NADP are used to drive the process leading to the synthesis of food, more accurately the sugars. That ATP and NADP is used for the formation of sugars or formation of the food, and oxygen that are released during the process is diffused out in the chloroplast. This is the biosynthetic phase of the photosynthesis. This process does not directly depend on the presence of light. This process does not directly depends on the presence of light, but it depends on the products of the light reaction, that is ATP and NADPH, beside carbon dioxide and hydrogen and water. You may wonder how this could be verified. It is simple. Immediately after the light become unavailable, the biosynthetic process continue for some time and then stops. If then light is made available, the synthesis start again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Here we stop the today's lecture, and now in the next lecture we see the dark reaction. Thank you.